So in Practical 1, the objective was to learn how to calculate and interpret descriptive statistics such as mean, variance and standard deviation, while also understanding the correlation. The material that we use is like leaves, a ruler and also graph paper. To collect 10 leaves from the same tree, then we measured their length and width and recorded the data. For each set of leaves, we calculate the mean, variance and standard deviation of their length and width. We repeated this process for 3 sets, which gave us data for 30 leaves in total. Lastly, using the data from all 30 leaves, we plotted a graph of leaf length versus leaf width to observe the relationship between these two variables. So here's the data that we got for Practical 1. From the graph, we observe that the data points are scattered, indicating no strong relationship between leaf length and width. For example, length may depend on cell elongation, a process driven by genetic factors and supported by hormones. Secondly, width on the other hand may rely on cell division, which is often more sensitive to environmental stress such as water supplies or light exposure. Lastly, leaf might prioritize either length or width depending on resource availability, which explains why this dimension vary independently.
Then, discuss the differences or similarity of the mean of length between different sets of leaves. Do the same for the mean of width. When comparing the variation between length and width, both parameters show some variation across the sets. However, the variation in width is slightly greater than in length. This suggests that leaf length is a more stable characteristic, while width is more dynamic and likely to respond to environmental or genetic factors. What is meant by variance? The leaves have different measurements of width and length. What is the meaning of this statement? Discuss with respect to variance. What is meant by standard deviation? Variance is a statistical measure that quantifies how much individual data points differ from the mean. The statement leaves have different width and length measurements. Standard deviation is another important measure that helps us interpret variance. Why is it necessary to have different sets of measurements for the measurement of leaf width and length? So using multiple steps allows for a more comprehensive understanding of variability. To study and analyze the diversity and distribution of plant species within a given area. Specifically, we aim to introduce the simple concept of quadrat to enhance the power of observation, to record the difference in plant distribution within the quadrat, and to infer from natural observations the possible factors that affect distributions and abundance of organisms. We use the following materials like raffia string, measurement tape, stick, scissors, notebook. Different types of forest at Taman Botanical Putrajaya will be the area under study. Second, make one quadrat of 5 meter times 5 meter at each forest type and take note of 3 stands that are found inside the quadrat. Measure the detail of brace height or TBH for each of the 3 stands within your quadrat. Data will be used to estimate the population size and population density. 5 plant species were observed and identified. These species had only one individual observed. The density for all species was consistently 100 individuals per square meter.
A quadrat is a frame used to study the distribution and abundance of organisms in habitat. The two key requirements for quadrat sampling are random placement to avoid bias and consistent size and shape for reliable comparisons. Larger quadrats provide better population representation but require more resources. Smaller quadrats are easier to handle but may result in limited representation and biased result. Diverse conditions and uneven organism distribution make accurate representation difficult. Thick vegetation, limited visibility, and species mobility hinder direct observation, while certain methods may over or underestimate densities. Variation in plant species across quadrats is influenced by macrohabitat difference such as soil, light and moisture, as well as competition and resource availability.